Bismillahirrahmanirrahim. 0610 IGCSE paper 2 March 23 MCQ. As you all know, in IGCSE the paper is only for 45 minutes. So I would say they're slightly easier questions because you're only given a minute for each question. So I find them slightly easier, but maybe you don't, as compared to 5090. Now, coming to question number 1, what is used as a means of classification? The number of strands of mRNA? No, mRNA is only when that protein is being made, then mRNA will be produced. The shape of the DNA molecule, it's not going to be any different. Is the content of the DNA, which is important. The sequence of bases in the DNA, yes. So if in the classification, this is the sequence in one animal, this could be the sequence in the other animal. So the thing is that it is the sequence of bases in the DNA which is used as a means of classification. The types of bases, why is that wrong? The reason why it's wrong, because types of bases are only 4 and every animal has ATGC. Every animal has ATGC. So it's not a type, there is no, but of course the numbers vary, matter how many percent is A and how many percent is C. That of course varies, but otherwise there are not many types of DNA. So that is why part D is wrong. Then comes question number 2, how many jointed legs? How many jointed legs does an insect have? Yeah, everybody knows it's 3 pairs. You look at a housefly, you look at a mosquito, they have 3 pairs, or you can say 6 legs. Then the micrograph shows a cell from a type of aquatic plant. Which labeled parts indicate that this is a plant cell? So basically it's going to be the cell wall, which is Q, and S is the chloroplast. So it is going to be, the answer is going to be D. So what is P? P is the nucleus. Nucleus is present even in animal cells. Nucleus is present even in fungal cells. And what is R? R looks like the cell membrane or the cytoplasm. More so, it looks like the cytoplasm because the arrow is going inside the cell wall. So cell membrane you cannot see in a photomicrograph. Cell membrane is it's only 5 nanometers in wide. So you really can't see it in a light microscope. And this is a photomicrograph, which means it's taken from a light microscope. Now, question number 4, the diagram shows a red blood cell. They've shown you the diameter is 49 millimeter. Look at the unit, 49 millimeter. The actual diameter of the cell is 7 micrometers. So first what you have to do is you have to convert 49 millimeter into micrometer. So this would be 49 and you add 3 zeros and this will become a micrometer. And then you have to divide it by 7 because 7 was the original. It says the diameter actually is 7 micrometers. So when you divide it by this 7, I know you all don't know mental math, you all can't do without a calculator. We are not brought up in that era. So that is why the answer is C. Which molecule crosses a partially permeable membrane during osmosis? Well, what is osmosis? Everybody knows in osmosis what happens. It's the water molecule. Question 6. The graph shows the rate of uptake of magnesium ions by two similar plants X and Y. The roots of each plant were placed in a range of solutions. Each solution contained a different concentration of magnesium ions. All other conditions were kept constant. So here we have, say, 1%, then 2%, then 3%, then 4, then 5, then 6, then 7, then 8. This is how you must do the MCQs. And then, of course, the rate of magnesium uptake. So in plant Y, what happens? The rate increases till, say, 5, and then the rate becomes constant. But in plant X, the rate increases more. The rate is more till, say, about 7 here, and then it remains constant. So here it was about, say, 5 here, and here it's about 7 here. And then the rate becomes constant, means the concentration of magnesium ions in the external solution in 7 concentration, the rate becomes constant. Now, what is the possible explanation of it? Now, before I give you the explanation, I want to explain active transport to you. What you have to understand is this is the cell membrane. So this is a phospholipid bilayer, and you study this in A levels in great detail. Now, what it has is that it has these channel proteins, and they have an ATP binding site. and the ATP is converted to ADP, and that provides it the energy. And then, of course, you see there are only three here, and there are 1, 2, 3, 4, 5, 6, 7 here. So they are moving in from low concentration to high concentration. So that requires energy. So this channel protein or this protein pump is going to pick up the molecule from this side and then throw it this side. Pick up the molecule from here and then throw it here. So what you have to understand, a cell membrane will have, say, only 5 channel proteins. But another cell has 7 channel proteins. 5, 6, 7, or maybe even more. 10 channel proteins. So you have to understand is the more the channel proteins, more the movement is going to take place, more can enter at a certain point. But then a time comes when all of them are being used and the ions are entering through those channel proteins. So the rate has to become constant. That is why the graph becomes constant then. The rate becomes constant. That means per unit time, say 10 molecules are entering. So the rate becomes constant then. Here the rate was not constant. Here it was increasing. Only one ion was entering, then two were entering, then three were entering, then four were entering. And a time comes when through all of them, ions are entering. Now they can't be any more. So this is a point which you have to remember. This is a point which they know that you don't know very well and they're going to examine this. So looking at this question, what is a possible explanation for the difference in results for the two plants? The answer is of course A. Plant Y has fewer protein carriers for magnesium ions in its cell membrane. Plant Y has a higher rate of respiration. It might have a higher rate of respiration, it might release more energy, but then if there are not enough channel proteins, how is it going to increase the rate? Plant Y has more root hair cells. It's the rate of uptake of magnesium ions. The root hair cells in plant Y have a lower water potential. Well, that's not going to affect the ion uptake. So it's a question of ion uptake of magnesium ions. A simple direct question, which substance is used to test a food for vitamin C? We all know that it's DCPIP. Then in DNA, what is the correct base pairing? You see, the base pairing is ATGC. I always tell you to remember it's at government college. So if it's either A here, or you can have the T here and the A here, or you can have the C here and the G here. So it's ATGC. It can't be anything else. ATGC. What is the correct base pairing? So T with G is wrong, T with T is wrong, T with C is wrong. It's always A and T, G and C. Whether the A is this side or that side doesn't matter, it is always like this. What remains at the end of an enzyme controlled reaction? What remains at the end of an enzyme controlled reaction? Yeah, it has to be A. Why? Because enzymes and products. All the substrates have been used up, the reaction is completed. So it has to be enzymes and products. Question number 10. Which feature explains the specificity of an enzyme? Complementary shape of active site and substrate. Yes, that is the answer. But why the others wrong? Well, we see all enzymes would denature at certain pH values. So that's not telling us the specificity. Specificity means that this enzyme, say this is amylase, why would it only work on starch? Because starch is complementary to the active site. Why would this amylase not work on a protein or work on a fat and convert the fat into fatty acids and glycerol? The reason is the shape of the active site and the substrate. But all enzymes denature at pH values. Temperature changes leading to change in enzyme activity. That's for all enzymes. And varying frequency of active collisions between enzyme and substrate. Again, that is something which is standard for all enzymes. But they've said what explains specificity of an enzyme is reading the English and understanding the English. Then coming to question number 11, a plant is placed in the dark until all its stored starch is used up. The plant is placed in light with black paper or part of one green leaf. Black paper over both sides of the leaf, hole cut in the paper. After 8 hours, the leaf is tested for starch. Which diagram shows the appearance of the leaf after the starch. And the starch present, this is, and then the starch not present is the clear area. The answer is B. Why is it B? Because you see this area, this area has to be green. Sorry, this area has to be black because that is the area which is exposed. So this is going to be the starch is present going to be here. It's going to be blue, black, or the iodine. And this area which was covered with light will not have any, covered with black paper, that will not have any starch in it. Because if light is not reaching it, no starch is going to be present in it. No photosynthesis, no glucose converted into starch. Question number 12. The diagram shows part of a cross section of a leaf. Which row shows the correct labels? Question number 12. Which row shows the correct labels? Well, what are the labels? You see, what you have to understand, what is R? R is the cuticle. S is the palisade mesophyll. T is the spongy mesophyll. So answer is A. Then coming to question number 13. The diagram shows the different types of human teeth. Which teeth are used for grinding food? Grinding food. Please do not write chewing food. We don't write chewing is not allowed. So the answer is A. Why is it A? Because 1 and 3. You see, this is the front tooth incisor and this is the canine. Now these are usually for cutting. So we're only left with 1 and 3, which are used for grinding of the food. So the answer is A. The diagram shows the villus structures P and Q absorb different products of digestion. Now what you have to understand is P is this part, the internal part. Sorry, Q. Q is this part and this is the lacteal. And lacteal, all the fats, lipids go into it. So Q has to be fatty acids and B has to be the other is the capillary network in which glucose, amino acids, vitamins, minerals all enter into the capillaries. So that is why the answer is C. Q has to be lacteals, lacteal has to be fatty acids, and it has to be P has to be glucose or could have been also amino acids, yes. But then this is wrong. That's why this is wrong, and that is why this is wrong. So the answer to 14 is C. Question number 15. The arrow show the movement of substance from the soil into the root hair cell. Which substances move from the soil into the root hair cell? From the soil into the root hair cells only water and mineral ions. You see, carbon dioxide and oxygen, why would that move in? There's no photosynthesis inside the root hair cell, so carbon dioxide is not needed. It won't diffuse in because the diffusion only takes place if there is a lower concentration inside the cell and a higher concentration outside. Glucose and water. Why would soil have glucose? So there can't be mineral ions and glucose. Again, that was wrong. Why would soil have any glucose in it? Question 16. By which process does water escape from the stomata in the leaves? You see, this is a leaf. And inside, first the water has to evaporate. So for the water to evaporate, it must form a water vapor. It doesn't come out in a liquid state. And then the water vapor is going to diffuse out. So that is why the answer is diffusion. Evaporation is only on the palisade and the spongy, mostly on the spongy mesophyll cells, when the spongy mesophyll cells have a film of water around it. And then this has to evaporate. So from a liquid state, it has to convert into a vapor state. And then the water vapors are going to diffuse out, which is called transpiration. So 16b. Which row about the circulatory system is correct? Part that pumps blood, part with valves. Question number 17. So question 17 has to be A, part that pumps blood has to be heart. So these are the only two possibilities. But here part with valves is heart and veins. Heart has valves, bicuspid, tricuspid valves, semilunar valves. Veins have valves also. Capillaries never have any valves. Yes, arteries do have valves, but then capillaries is the wrong one. So that is why 17A. Then 18. The graph shows pressure changes that take place in the right atrium, right ventricle, and left ventricle of a human heart when the muscle wall contracts and relaxes. What is the pressure of the right ventricle when the left ventricle is at its maximum pressure? Question 18. The graph shows pressure changes that take place in the right atrium, right ventricle, and left ventricle of a human heart when the muscles will contract and relax. Now you know greater pressure, so this would be the left ventricle. This would be the right ventricle, and this would be the right atrium. Because you see, the pressure, ventricular pressure is the highest. Ventricle have the highest pressure because left ventricle has a very thick muscle, so it will have a lot of pressure. Now it says, what is the pressure in the right ventricle when the left ventricle is at its maximum pressure? Now, so this is the left ventricle maximum pressure. Now this is the point when it's about 3. So the answer is C. A difficult question, I must say, it's a difficult question. Question number 19, the sequence of amino acids and antibodies enable them to complete which function? Bind to a specific antigen, antibodies. Remember antibodies, lymphocytes make antibodies, they attach. So this is a virus and this has got this antigen on it. So there will be this antibody which is going to attach to it. Bind to all pathogens, perform phagocytosis, confer passive immunity for all diseases. Yes, everybody knows that they bind to antigens. So question number 19, the answer is A. Question number 20, the table shows some of the changes that occur during breathing. Diaphragm P from contracted to relaxed, X from relaxed to contracted, external intercostal muscle Q and Y, internal R and Z. Which changes occur to cause inspiration? Inspiration means inhalation. So you have to understand is in inhalation, the diaphragm has to go down. The external intercostal muscles have to contract to pull the rib cage upwards and outwards. The internal intercostal muscles has to relax. So all these situations, if you know, only then you can do this question. So what we have to understand is that the answer to this would be what? X, Y, because the diaphragm from relaxed to contract, diaphragm has to contract and become flattened. External intercostal muscles have to contract as well. But internal intercostal muscles must relax. So it is X, Y, and R. So the answer is C. Why? Because internal intercostal muscles only work when we are forcefully exhaling, like when you're coughing. When you cough, you forcefully exhale, and that is when the internal intercostal muscles come into play. So that is the answer to question number 20. Now we will continue this in the next video, the rest of the questions, and I hope this is all going to be helpful for you in the upcoming exam. Thank you very much, and thank you for subscribing. Thanks you, everybody.